I'm Shamima Rahman. I'm a consultant at Great Ormond Street Hospital and the Institute of Child Health, which is part of University College London. And I know some of you, but not all of you. And today I've been asked by Alison to do something that she calls a research update. And I had to call her to get clarification or email her because I wasn't sure what she meant. And she thought that you really wanted to know more about new treatments. But I'm going to start with a brief genetics update as well, because it's very relevant to new treatments, as you'll hear. And the genetics, I'm going to talk about whole exome sequencing and whole genome sequencing. And if you haven't heard of those before, don't worry, I'll explain them in a moment. I know you have a genetics lecture, so I won't dwell too much on it. And then most of this is going to be about emerging therapies, strategies for developing novel treatments, pharmacological approaches, and genetic approaches is the other big group. And then I'm going to talk about clinical trials. Oops. Both clinical trials that have already been done in the past and then trials that are ongoing and what results there are. Those are things you thought we'd cover. I need to have a door monitor. I wonder if someone would mind sitting in that seat and opening the door for me, please. Thank you, Karen. So in terms of genetics and mitochondrial disease, the reason why I think genetics is critically important to treatment is that if you don't know what it is you have, then you can't possibly hope to treat it properly, and I hope you'd all agree with that. So until very recently, we struggled to make a genetic diagnosis for most people with mitochondrial disease. Maybe 10 or 20% would get a diagnosis, and for the rest we just didn't know. And then about 5 years ago, we moved on to using things called next generation sequencing technologies, by which instead of just sequencing one gene at a time, you could sequence all the genes in our cells, so all 20,000 of them. And the whole exome approach is where we do the coding parts of all 20,000 genes. And we did a research study where we looked at 4 groups, either children with complex 1 deficiency, or complex 4 deficiency, or several enzymes being low, or normal enzymes, but I was absolutely convinced they had mitochondrial disease. And across the board, whichever group they fell into, we found an answer of between 50 and 60%, so almost a 5 to 6 times increase on what we could do before. And one of the things that was really baffling, because we looked at about 100 families, and each family had a different gene, so that began to worry me that we were going to have our work cut out trying to treat mitochondrial disease. Now, 50-60% probably doesn't sound great to all of you. I was pleased that it was better than it had been, but it should be 100%. Why aren't we finding the gene for everyone? And it's really complicated, and sometimes it's because even though these new technologies are so much better than they used to be, they're not perfect, and maybe the gene that we want hasn't been captured, or there's a sequencing error, or there's something called a de novo mutation. We assume for mitochondrial disease that most are recessively inherited, where the parents are carriers and the child has got 2 copies, both copies don't work properly. And because research funds are tight, in my research study, just looked at the affected child. And if there is, on the other hand, a new change that's only in the child and not in the parents, and 1 copy is enough to cause disease, we'd miss it in that model. And so to try and avoid that, you'd have to sequence both parents and the child. And there are some other reasons why you might not find the answer. So there might be a big bit of a gene missing rather than a little point change, and these new technologies aren't as good at big bits missing. The other thing is that it may not be primary mitochondrial disease, but masquerading as mitochondrial disease. And if that's what it is, then it's very difficult on the assumption that it is mitochondrial disease to find the right gene. So we've talked about exome sequencing, and then in the last couple of years we've been very lucky in that David Cameron, who himself had a child with a rare disease, has thought it's very important to invest in finding answers for families affected by rare genetic diseases, and so has introduced something called the whole genome sequencing study for 100,000 families, so the 100,000 Genomes Project. At UCL, we were one of the big centres doing the pilot study with Genomics England, and we recruited about 2,000 into that study, including 40 families with mitochondrial disease. The thing with a whole genome is it is much bigger than an exome. Although an exome covers all the genes, it's only the coding parts, and to give you an order of magnitude, an exome is only about 2% of a genome. So the bioinformatics around understanding the genome has been very complicated, and we're only just about to get, we hope very soon, the data from that pilot study of the Genomics England project. And again, to just give you an idea of scale, when we only look at one gene at a time, we only expect to find 1 or 2 changes, and they may or may not be disease-causing. However, when we did the exome, we know that we found at least 20,000 changes, even in people who are perfectly healthy. And then we had to try and sift down to find the 2 changes that were causing disease in that person. And sometimes you use really good exome sequencing, you can find as many as 50,000 changes. And I've told you how much bigger a genome is, so we expect to find 3.5 million variants in each of the genomes. So to try and find the needle in the haystack is going to be very challenging, and that's why it's taken so long to get the results back from that study. The good news is that the families with mitochondrial disease were so great at volunteering to be in the pilot study that Genomics England are very happy to keep mitochondrial disease in the main study, and we are recruiting at Great Ormond Street and Newcastle and other centres for the study. So if you don't have a genetic diagnosis and you're not already in an exome study such as the Evelina Lilly exome study or the DDD study, or you already know that those studies were negative, then let us know in your local centre and you can be recruited to that study. I finished on genetics unless there were any questions about genetics. It's amazing, having the camera here has silenced you all. The last lot were much more talkative. So I'm going to move on to treatment. And the question that I'm sure has been on the tip of everyone's tongue in this room, even Professor Taylor in the back there, is why is it so difficult to treat mitochondrial disease? And the answer to that is very complicated, but the genetics that I've told you about is part of that. So we already know that there are more than 200 different genes that can cause mitochondrial disease. And if there are that many different diseases, and possibly the real total is going to be more like 1,000 or even 1,500, I think it'd be a bit naive to think a single treatment could treat 1,000 different diseases. So that's a problem. Another thing is that, as you know, sadly, often when you have mitochondrial disease, it's not just one part of your body that's affected, but it can be lots of different areas of the body. And again, to expect a treatment to sort everything out is perhaps unrealistic. Thirdly, the mitochondria are tucked deep inside cells, so to get medicines and genes into the mitochondria to correct the defects is very difficult, very challenging. They've got to cross 2 membranes that bound the mitochondrion. And the very last thing I wanted to mention is that historically we didn't have any good animal models for mitochondrial disease. You may be wondering why not if that matters for treatment. It does matter because when we do clinical trials, there are a phase of things that happen, a series of things, and the first is a preclinical trial, and a preclinical trial is usually done in an animal model, often a mouse or a rat. And I don't know if some of you are against animal testing, but sometimes for really terrible rare diseases, I think it's unavoidable that we do have to do that. It is done in a compassionate way, but it's the only way to often to get permission to go on to the human studies. Particularly something's a dangerous drug, I think you'd rather it was found out in an animal model than waiting until it's tested on people. So after the preclinical studies, then there's usually a phase 1 study in people, which is a dose finding or safety study, and then there's phase 2 and phase 3, which are often randomized, double-blind. I'll explain what those mean as we go on. So that's why animal models were important. I've talked about the complexity of mitochondrial disease. So we know about the clinical complexity that any part of the body can be affected in any combination. The cartoon on the right shows just some of the reactions that are going on in the mitochondria. They're very complex organelles. So there's a lot of different biochemistry going on there, and you may need different treatments to treat different things that could go wrong. And then I've talked about the genetic complexity with the mitochondrial genome at the bottom left, and then the nuclear genes, the whole genome on the right there. As we found out more about the genes that cause mitochondrial disease, we've seen that there are very many different disease mechanisms, and we're still trying to understand them. There can be a problem with the mitochondrial DNA itself, or in maintaining or replicating it, or in converting the information stored in those mitochondrial genes into proteins, which are all part of this OXPHOS enzyme system of 5 enzymes there. there can be problems with importing chemicals or proteins into the mitochondrion to do their job, or there can be a problem with the wall of the mitochondria themselves, or in the movement. Mitochondria aren't static organelles, they're constantly moving, they're joining up in a process of fusion and then dividing in a process of fission, and those things can go wrong. Or it may be in one of the vitamin cofactors that we need to help build these oxphos complexes, such as coenzyme Q10, but there are many other cofactors that we need. So putting all of that together, this is a slightly old slide, but we know of more than 200 genes that can cause this. And in the middle of all of that massive information, there are a few genes where we know that if we get in there early enough, they're potentially treatable conditions. So the first thing I want to mention to you is that there are a few treatable mitochondrial diseases, and we're building up this jigsaw puzzle. There are only a few pieces here at the moment, but I hope that this is going to need smaller pieces and be a much bigger jigsaw as time goes on. So the first thing is that some people cannot make coenzyme Q10, and if that's the problem, then often giving very large doses of coenzyme Q10 can circumvent that problem. The next thing is that some people are responsive to another vitamin, riboflavin, so a B vitamin. And one of those is a type of complex 1 deficiency called ACAD9 deficiency. And if that's the case, we know that giving a good amount of riboflavin would help. The other thing is that the more time I spend in a mitochondrial clinic, the more I realise there are lots of other conditions that can mimic mitochondrial disease. And some of the patients that I've treated with riboflavin have got better, not because they have primary mitochondrial disease, but they have a problem with getting riboflavin into the right part of their body, into the brain. And these are called Brown-Violetto-Van Leeuwen syndrome, the BVVLS that I've indicated there. And so I've treated quite a few patients with that condition now, with very high doses of riboflavin. There are two other conditions that can be responsive to a vitamin called biotin. There's biotinidase deficiency, and everyone that we see at Great Ormond Street with suspected mitochondrial disease or lactic acidosis or any kind of neurological disease, particularly in the first year of life, we often will measure the biotinidase. It's just a simple blood test, and we might give biotin until we get the result of that back. There's a different condition that we initially knew of in Saudi Arabia called biotin-thiamine responsive basal ganglia disease, and we thought it was very rare and caused an adult onset disease, but we've now realised it's one of the causes of Lee syndrome. And there you need both biotin and thiamine for effective treatment, and usually making a genetic diagnosis is the clue to treating that condition well. And then the fourth group, there isn't a specific treatment as such, but they get better by themselves. They have what we call benign reversible mitochondrial disease. It's not that benign because they might need a lot of support, such as being ventilated for many months or having a liver transplant, but we are always, when we first meet someone with mitochondrial disease, wondering about the possibility of one of these four conditions and making sure that we've excluded them with a test or that we've tried the treatment. The other thing is I know there are other talks that are mentioning supportive therapies, but I want to mention this because this is a talk about treatments. And when people tell me there are no treatments for mitochondrial disease, I feel quite sad because there are treatments, they're not specific curative treatments. So we would always look for all the things that we can treat. So seizures with anticonvulsants, ptosis with eye surgery, cochlear implants or hearing aids for hearing loss, and so on and so forth, and going up to and including in some patients, depending on whether it's the right thing for that patient, even a heart transplant or a kidney transplant. I'm going to move on to drugs. We have here a whole series of medicines that I've been asked about in my clinic over the years, and I know in America some doctors like cocktails and they give all of these together. I personally don't, because I'd like to know what it is that's helping someone. I don't want my patients to rattle because I've got all of these things inside them. So moving on from those, why are all these medicines out there as possibilities that you've read about on a website or in a paper or something, or someone's mentioned to you? And why am I going to carry on talking more and more about clinical trials as this talk goes on? I'm going to use something called Lee syndrome, which I'm afraid that probably quite a lot of you are familiar with, as an example here. So when someone has Lee syndrome, it's usually a baby, and they've often been completely normal. This is kind of development skills along this y-axis, and this is time along here. And the first illness or decompensation lose skills, which is a terrifying thing to happen to you as parents. And that's probably the time when the diagnosis is being thought of, may be made, may not be made, but certainly interventions are not really happening at that point. Get better by themselves, even up to the skill level of before, but not up to the trajectory that they were on before. And then another episode of decompensation happens here. And then no doctor, no parent is going to want to see a child losing skills and not try and do something to help. And they may try one of those medicines I've shown you, they may do something else, tube feeds or something, or they may just give some rhubarb, I don't know, I'm picking rhubarb as a crazy example. And as before, things get better, maybe because of the rhubarb, or maybe it was spontaneous, as here. And then, because we're always trying to train people and junior doctors like to write papers, somebody writes a case report that gets published in a journal saying, rhubarb cures Lee syndrome. And it's out there. Rob's smiling at the back. And so then, maybe the child has another decompensation while still taking rhubarb. But I'm an editor of a journal. I know how difficult it is to publish negative papers, and no one ever writes a report to say, actually, rhubarb isn't that good in Lee syndrome. And that's why all of these things are out there as possible treatments, and people swear by them or not. And that's why we need to have clinical trials, because you really need to compare rhubarb to something else or nothing and see in a large number of patients to see if it actually does make a difference. So evidence is becoming an important concept I'm telling you about, and one way of collecting evidence is through clinical trials, and there are Cochrane systematic reviews, and colleagues of Professor Taylor's and people in Finland and in Australia did something called a Cochrane systematic review, and what they did was they took more than 1,000 papers writing about mitochondrial disease treatment and looked at them, to evaluate them to see if they were good clinical trials. And out of that more than 1,000, they only found 12 that they thought were good clinical trials for mitochondrial disease, which is very disappointing. I think it's 1% or something of that. And what they concluded, having looked at those 12 trials, is that they even contradicted each other, and they concluded there was no evidence for or against any of the treatments that had been tried in those trials, and that further research was needed. And the thing that's really sad is that that's the same conclusion that was reached when they last did that exercise in 2006, so 5 years later, still no really good trials. What are those 12 trials? Some of you may have seen, I left a couple of handouts, and if you didn't manage to pick up a handout, Alison can email them to you, or my secretary's email is on the flip chart over there, and you can email her and she can send them to you. They are written in medical speak, they weren't written for parents, and I'm sorry, but they do contain this table. And this is a list of those 12 treatments, those clinical trials, and there are only the 3 that I've ringed here that had more than 30 people in them. Now, this is a problem, because if you were, say, working in heart attacks and doing treatment trials, you'd have 4,000 people in your trial. So to have statistics that work for 30 is challenging. And actually some of these only had 5 in, so that's difficult. And coenzyme Q10 was looked at in this one with 30, and it was a good trial design. It was randomized, it was placebo-controlled, double-blinded, crossover, I'll explain all of that later. And they concluded that maybe it helped the lactate levels a little bit, and maybe they were a bit better after doing bicycle exercise, but no significant change in other things. And this one down here also had CoQ as part of a kind of cocktail with 2 other things. And they said maybe the lactate was a little bit lower, and maybe there was a bit more muscle strength. But they weren't major significant findings. But because of those 2 trials, one of the reasons why I think at least coenzyme Q is probably safe, and I know it's a good antioxidant, that's why I tend to use it. I'm moving on now away from what clinical trials have been done, back to the preclinical phase. So we're back to the mice studies. And I'm going to look at 2 groups. One is the pharmacological approaches, or medicines, and the other is gene therapy. So this is the Oxford system again with the 5 enzymes. And your child may have one of those enzymes deficient or all of them, or maybe not a measurable defect in those. But we know that as soon as something goes wrong in that system, then things called reactive oxygen species build up. And and that's because we're using oxygen and food in the mitochondria to try and make energy. And so all of the antioxidant medicines aim to try and neutralize these, as it were. And those antioxidant medicines include coenzyme Q and variants of, including something called EPI 743, which I'll come back to, vitamin E, vitamin C, something called Trolox, and also something that's known as RP103 in the clinical trial literature. And again, I'll come back to that. And so far, there isn't any evidence for benefit or harm from any of these conditions. And so that's why people do tend to use coenzyme Q unless you're in a trial of one of the other treatments. So that is targeting reactive oxygen species as the first strategy. The next thing is we know the mitochondria aren't working properly, so wouldn't a good idea be just to make more mitochondria? And so a lot of strategies are looking at that. Now, making mitochondria is a pretty complicated process. This is the cell nucleus, and there are lots of different, what we call transcription factors, that are involved in coding for different parts of the mitochondria to be upregulated so you can make more mitochondria. But if you look along the blue bar here, these are some of the things that impact on this whole system of transcription factors. So there's exercise, and then 3 medicines, ACAR, resveratrol, and bezafibrate. The last thing is caloric restriction. I know that most of you with affected children will know that getting any weight on them is really hard, and the last thing you want to do is to give them a caloric restriction. I wouldn't recommend it at all, because you've already shown from nature that your child not getting enough calories is not helping them get better from their mitochondrial disease. But that's kind of part of the background to the ketogenic diet and why people thought it might work. Again, there's no evidence for it there. The only thing that we definitely use ketogenic diet for is PDH deficiency, where we think it works for a different reason. In PDH deficiency, there's difficulty in getting fuels to the brain, and ketones are an alternative fuel source. So how can we make more mitochondria? How can we stimulate this process of mitochondrial biogenesis? Well, let's take 3 examples, the bezafibrate medicine, resveratrol and ACAR, and they've all been tried in different cell models of mitochondrial disease and mouse models. Now, I'm going to focus mostly on bezafibrate. It's been tried in 3 different mouse models of mitochondrial disease, and sadly, they saw different responses in all 3 sets of mice, even though they all have mitochondrial disease. The other thing is that this is an example of why we can't just do mice studies, because we know that bezafibrate is quite a safe medicine because it's used in lots of people with heart disease, with high cholesterol, to try and bring cholesterol down. And mice don't have the same metabolism as us, so mice with bezafibrate get liver disease, fatty liver disease, so it's not good for them in the longer term, but their muscle did seem to get better. So people have done a study of bezafibrate in a different kind of mitochondrial disease. The mitochondria are involved in breaking down fats by a process called fat oxidation, and people who can't do fat oxidation properly have exercise intolerance. So they've done a study, a randomized, double-blind, crossover study of bezafibrate in fat oxidation defects, and it made absolutely no difference. So the mice can work and then people can't. So I know bezafibrate was one of the medicines I was asked about for this talk, and I'm not aware that, unless you are, Prof. Taylor, no, of any human studies for bezafibrate in mitochondrial disease at the moment.
There was an abstract at the meeting you were at last week.
Did I not read it? Yeah.
They have tried it in, that's the group from Salzburg.
Okay.
I don't know anything.
They didn't talk about it, no. So and because of the disappointing results in the fat oxidation, which would work by the same mechanism, I don't think that it's still the great hope for treating mitochondrial disease. Resveratrol is a substance from red wine, which helps to increase mitochondrial biogenesis, and it's being trialled in Friedreich's ataxia, which is a genetic condition that causes mitochondrial dysfunction secondarily. And then ACAR is also being tried. So those are the mitochondrial biogenesis stimulators. Recently, there's been a new medicine, which is a form of vitamin B3, nicotinamide riboside, which was shown in aging studies to increase mitochondrial biogenesis. And what it does is that it replaces something called NAD+, and that's here. And NAD+ gets used up when the mitochondria aren't working properly, and it can cause things to stall, and so it's a good idea to try and replace that. And that's been trialled again in a few mouse models, and it looks like there are encouraging results, and there's been talk about human clinical trials, and again, I don't think anything has started yet. Nope. Rob's shaking his head at the back. So I've now finished on the pharmacological studies, unless you have any questions about any of those medicines. I'm going to go on to gene therapy then. So, we've talked about how there are two genomes involved in mitochondrial disease, and I'm going to start with the mitochondrial genome, which is small, but it's tucked right inside the mitochondria, so it's difficult to get at. It's also got a few other weird things. It's maternally inherited. Now, genes, DNA, consists of 3-letter sequences, and each 3 letters codes for an amino acid, and that's called the genetic code. And because mitochondria are derived from ancient bacteria, they have a different genetic code to our nucleus, and that's going to be important in something I'm going to tell you about in a moment. So just remember that. They're also present in many, many copies in our cells, which means that we talk about the heteroplasmy or homoplasmy. Homoplasmy is where all the copies have mutation, and heteroplasmy is where a percentage do. And for most diseases, it's more than 50%, maybe even more than 70%, but it depends on the mutation and the disease. The other thing about mitochondrial DNA is that there are already more than 250 mutations, so you'd maybe have to have a different gene therapy for each single mutation unless you can find a generic strategy. So I'm going to talk to you about some of the experimental ways of trying to cure mitochondrial DNA that are out there. One is that we know that there are enzymes whose job it is to cut DNA in particular places, and so if you could get those enzymes into the mitochondrion, maybe that would work. And you could just send them in, as they're called restriction endonucleases as themselves, or they can go packaged up in something called zinc finger nucleases, or the latest way that's been quite exciting is the mitotalons. And here, and a related technique called CRISPR, a guide wire is used to get the DNA into the right, the enzyme into the right place, so it can cut the DNA at a specific point. And they've already shown in cell models, and are now working on mouse models, that the mitochondrial DNA deletion that causes Pearson and Kearns-Sayre syndrome can be selectively removed, and the healthy, normal, wild-type DNA can take over using this method. So that is quite exciting, but they now need to scale it up from cells in culture and little mice to whole people. But it's definitely something that is looking potentially promising. The next thing is, I told you about the weird genetic code in the mitochondrion. So one way is if you have a mutation in one of the 13 mitochondrial genes that codes for a protein, you could recode it into the nuclear gene code and then send it into the nucleus and have it re-express and make the protein inside outside the mitochondrion instead of inside it, and then get it imported. And that sounds fanciful. I wish I'd thought of it. It's very clever. And that has worked in a rat model of mitochondrial disease called Leber's hereditary optic neuropathy, which is caused by mutations in subunits of complex 1. And it looked very effective. And there are, I think, 3 different human clinical trials of this allotopic gene therapy, allotopic expression gene therapy going on for Leber's hereditary optic neuropathy. The next thing is trans-kingdom gene therapy. This is taking the even a further step out. So we know that there are those enzymes I've shown you in the mitochondrion, but other organisms have different enzymes that do the same job that we don't have as humans. So if you could get those, say, from fly or worm or some other organism, their alternative oxidases into our mitochondria, maybe you could use them as bypass therapy. So one called NDI could be used to bypass complex 1. Another called AOX, or alternative oxidase, could be used to bypass a complex 3 or complex 4 deficiency. And so these are all being looked at a very fundamental research level. They're not anywhere near towards a clinical treatment. Now, Newcastle are here enforcing, there's a talk about this, I'm not going to say very much, but another way of preventing mitochondrial DNA disease, which isn't gene therapy, but it is to prevent the transmission of the mitochondrial DNA in the egg or the embryo. So there's 2 techniques, pronuclear transfer or metaphase spindle transfer, and they've been shown to be effective both in human eggs that were damaged and therefore could be, and embryos that could be used for research, and also in Oregon in several rhesus monkeys have been made by that technique. And I'm sure that many of you will be familiar with the debate that's gone on in the House of Commons, House of Lords, and the legislation, and these techniques are about to be licensed in clinics from the end of October. I think the applications go in. So I've talked about treating mitochondrial DNA disease. The nuclear genes are actually relatively easier to do gene therapy for if you know what they are and if you have enough patients, because we've already got experience. My centre at Great Ormond Street Institute of Child Health, we've been doing gene therapy for immune deficiencies for more than a decade now. For example, for severe combined immune deficiency. And the idea of gene therapy is that you use a virus, most commonly a lentivirus or something called adeno-associated virus, or AAV, to get to carry the gene into the nucleus where it's expressed. And so if you have lots of the good copy of the gene, you're going to make normal protein, and that will kind of dilute out the defective protein that's made from the mutated DNA. So that's been tried for 2 mouse models of mitochondrial disease.
Someone received a text from one of the children.
Ian dot. Sorry.
And so they use the lentivirus for a disease called Mingy, or mitochondrial neurogastrointestinal encephalopathy. And because the enzyme that's deficient in that condition, called thymidine phosphorylase, is in blood cells, they put the lentivirus into the blood of the mice, and they were able to show biochemical correction. Whether this will lead to sustained permanent correction in people needs to be looked at in a clinical trial. And then ethyl-malonic encephalopathy is another mitochondrial disease causing complex 4 deficiency, and there they used AAV to carry the gene in. Now, the EME gene is called ETH1. There's a lot of it in the liver, and so they, because mice are little, it's hard to get the gene to the right place, so they did an intracardiac injection of the gene. It's not something that we'd think of for children or adults. It was just the way in the mice to get it into the liver, and that also showed biochemical correction. And these mice, instead of dying at a month of age, lived to more than 6 to 8 months, where they had to stop the study. So that all looks encouraging. And then I was once asked this by a dad in clinic, and I thought it was a very smart question, but I thought it was a kind of a crazy question, which was, why couldn't you just put more mitochondria into the cell? And people in the world are actually looking at that as a possibility, just transfusing in mitochondria. So those are the research techniques I wanted to tell you about. And I wanted to just finish that research bit on this. This is a database called PubMed, where publications are catalogued. And if you look, the first mitochondrial DNA mutations were found in 88, and that first decade was more about genetics. So my search term was mitochondrial disease treatment. And there were fewer, 100 or fewer papers for the first 10 or more years. But can you see there's been an exponential rise in papers on mitochondrial disease treatment, and I hope that that's going to be followed through with more and more clinical trials.
What's the significant drop there?
It's because I didn't make that slide today. Most of my slides I've made for this talk, but this is one I made for some doctors last year, so that's why it's cut off halfway through 2000. This is a talk I gave in September 2014, and I just couldn't bring myself to do it again yesterday. I should have done. And so I imagine it did carry on going up. I'm sure, in fact, I know for a fact that it did carry on going up. So all very well doing all this research, but we need to get it into the clinic. And how are we going to do that? And what's stopping us? Well, I hope you all agree with me now, well, we need clinical trials, but the ideal one needs to be adequately powered. And the power depends on the size of people in it and also the effect size. Are you going to see a 10% improvement, 100% improvement? And it needs to be statistically valid. It needs to be not a chance finding. I'm sure none of you want us to identify a drug as being effective because we made a mistake with our stats and it was just a chance effect. It needs to be randomized, it needs to be double-blinded. That means, so randomization is where a computer decides whether you're on treatment A or treatment B. Double-blinded means that neither the patient nor the doctor knows what treatment you're on. And that's really important because we as doctors want our patients to get better. And if we know they're on the active arm of the trial, we might kind of, not consciously, but subconsciously, you could introduce a bias towards thinking they were better. And it needs to be placebo-controlled, ideally, although in some situations it's very difficult to want your child in a placebo is nothing active in it. And in that case, we might want to compare to a historical treatment or have two different treatments and then cross them over. That's what a crossover study is. And then the last bit might make you smile or cry, I'm not sure which, probably the latter. An ideal trial needs a large group of patients with the same genetic defect, and if it's mitochondrial DNA, the same amount of mutation, with the same clinical problems and the same biochemical findings and at the same stage of disease. And you're shaking your head and you are smiling because you know that if we did a straw poll in this room, I think every single family in this room would have a different gene, a different biochemistry, a different set of problems and be a different stage. And if the Lilly Foundation cannot bring together a set of people for a clinical trial, then nobody in this country can. So what are we going to do about it? So, well, we need to think about different ways for rare diseases of handling the clinical and genetic heterogeneity in the small numbers and do unconventional clinical trials. We also need to know what the natural history of these diseases is because people aren't going to want to be on placebo arms, so we need to know what happened before any treatments came along. And we also need to work on outcome measures. What are we going to measure as the evidence that a treatment worked? So this slide shows that we are working internationally. These are all the national and just some of the national and international consortia that have been developed because we know that if you've got a really rare disease, that there are only 3 families in the world, one in Britain, one in Australia and one in Canada with that gene, then you're going to want the whole world to work together to try and find a cure for your gene, and that's what we are all trying to do. It's difficult, but I've got a telecall to NIH on Monday evening for exactly that reason, talking about what we're going to do next. I have been very lucky in that I've worked in the same centre for more than 20 years and I've seen a lot of people with mitochondrial disease, and I don't want it to be something that's in my head and no one else knows. So we've been trying to put out natural history studies, and this is an example for Lee syndrome, and there are the 3 lines are 3 different genes, and the shallower the slope, the slightly less severe. And can you see that this one is less severe than these 2 in the early parts of the disease? And so you don't want to be giving a treatment to someone with this gene and comparing it to that data because you might think you've found a miracle cure and actually you haven't. You're just looking at the wrong set of natural history data. And the same for people with mitochondrial DNA deletions. We've shown that if you've got Pearson syndrome or Kern-Sayre, or if you've presented in adulthood rather than childhood, you've got a very different slope of that graph. So it's important that we have that data. We also are looking at outcome measures. Should we measure lactate? Should we measure it in blood, in spinal fluid? Possibly not the best outcome measure because a lot of people don't have high lactate even though they have mitochondrial disease. And also as you get older, even if you had a high lactate earlier in life, it might not be so high. In the adult world, they often do serial muscle biopsies. I really wouldn't advocate that as something to do for children. I think it's far too invasive as a outcome measure. Again, in adults it might work if you've got brain imaging changes and you can follow them serially and they've got a chance of getting better. But again, the trouble is most children can't lie still for an MRI scan, and so again it may not be the best outcome measure. And there are new blood biomarkers such as FGF21, and we're looking at those. And then often it's going to be something like a composite score, such as GAIT or the Newcastle Pediatric Mitochondrial Disease Score, or is that Nijmegen? One of those, and then for Nijmegen or Newcastle. And those are going to be really important, how we measure the outcome. These are some of the clinical trials in the clinicaltrials.gov page that have got any relevance to mitochondrial disease. And I know you wanted an update, and I've been busy badgering the companies who are running some of these clinical trials for updates, and I have a couple for you. These are hot off the press. They were sent to me about midnight last night, so I couldn't have got you fresher information. So Matt Klein, who's the Chief Medical Officer of Edison Pharmaceuticals, this is a direct quote in his email. They are at the final stages of analysing all the clinical and biomarker data for the Lee syndrome EPI 743 trial, and they hope to make an announcement in the next 6 to 8 weeks. They didn't give me any clue. I asked them a specific question, how many patients were in the trial, and they wouldn't tell me, and they wouldn't give me any indication of what they're going to announce. But they are planning to launch another wave of studies. And RP103, which is a cysteamine, that's a phase 1 study, so it's dose finding and safety, and that's recruiting in 4 centres in America. I hope what I've shown you is that I think we need to move from a phase where we have genetics, whole genome or whole exome sequencing embedded in the NHS, and that's the aim of the 100,000 genome study, and that we then can translate the research findings to getting a really tailored treatment for your child. So we have individualised genomic medicine in the future as a hope. To summarise what I said, I'm really sorry to have to tell you that for most people with mitochondrial disease, there still aren't any curative treatments. But despite the very many barriers, there are very many innovative treatments that are being developed in the preclinical stage, both pharmacological and genetic. And the most promising pharmacological strategies are either antioxidants or aiming to increase mitochondrial biogenesis. Lots of gene therapy methods are also being explored, and most novel treatments, as I've said, are still at the preclinical stage. I'd like to thank you all for listening in this really hot room.